So a quick announcement. Today we have a conference dinner at 7 p.m. at this restaurant, Bovino's Fast Grill. This is the address, Alameda Santos 2393. It basically has all you can eat food plus a non-alcoholic beverage. And it's free, okay? It's paid by the Institute. Yes, that's all. Thank you. Yes, of course. You can pay separately for alcohol. You should, maybe. No, I don't know.
So let's start the section this afternoon. And we have the pleasure to have Karin speaking to us again on her second talk. Now she's going to talk about biomimetic membranes as a platform for the study of antimicrobial peptides. So Karin, please, your stage.
Aqui. E o colar é só... Aqui aperta. Isso. E aqui, acho que você desligou. Ah, então ele estava ligado. Pronto. Agora sim. So, yesterday I talked more like general stuff, what biomembranes are, not biomembranes, but these model membranes, what we can do, the techniques that we use. And tomorrow I'm going to show an example of an application of that, especially to study the interaction of these model membranes with a specific class of molecules, which are these antimicrobial peptides. So I'm going to use several of the techniques that I showed yesterday, and of course the lipid bilayers, which are like the center of my study. So the idea behind now, so the interest to study that, and this is not working here, is because, as you all know, there is a growing issue about antimicrobial resistance. So with all the pills that we get, conventional antibiotics, so the bacteria become resistant to that, and you have to find new ways to kill the bacteria. No? So is dangerously increasing, no? And so we need something new. And these antimicrobial peptides are one solution. So it's a way that people are thinking to overcome this problem. So what are these antimicrobial peptides? So originally, they are components of the innate immune system of us, of any animals, plants, even bacteria have some antimicrobial peptides. So this, but because we found this in the nature, then of course we also started synthesizing new compounds based on that. So apart from the natural sequences, we also use synthetically designed antimicrobial peptides. But usually they all share some characteristics. So peptides are small proteins, no? So they are typically 10 to 40 amino acids. No? And especially they have several basic, so cationic amino acids and hydrophobic amino acids. So this is very important for their action. So this means that they will have a net cationic charge, typically 3 to 10 plus charges, and they are amphipathic. No? So because they have charged and hydrophobic amino acids, they are amphipathic. So if you remember, the membrane is amphipathic, so it means that they will naturally have an affinity for the membrane and naturally an affinity for the membrane of microorganisms which are negatively charged. So they are very different in shape, and so they can have various structures. Oh, sorry, this thing here is not working. Saco. So they can have various structures, no? But most of them, when they are like acquire this secondary structure, they have this amphipathic character. So the charged amino acids are on one side and the hydrophobic are on the other side. So they have this ability to kill microorganisms, especially bacteria and fungi. And this is because they have a non-specific interaction with their membrane. So they insert into the membrane, and they can either form a pore, which we call a toroidal pore. So they make opening of holes in the membrane, pores, or there is also this carpet mechanism in which something more like a detergent-like or membrane disruption events occur. And of course, if you do that to the membrane of any living organism, it will lead to the death of this organism. And for the bacteria, it's more difficult to become resistant to that because it's something very basic. So the bacteria has its membrane. It cannot change and have a non-amphipathic membrane, for instance. So if we look at the bacterial membrane, you know that there are gram-positive and gram-negative bacteria. They are structurally different. So the gram-positive have one bilayer. The gram-negative has two bilayers. One is very specific. Very specific with these lipopolysaccharides outside. But what is relevant that I told you is that even these membranes here, they are full of anionic phospholipids, so especially this phosphatidylglycerol and cardiolipin. So that's why it's interesting to use the model membranes that I showed you yesterday. And we have them making lipid vesicles, liposomes. And if you remember my talk from yesterday, I told you about this different in size. So there are the small ones, well, I mean large compared to the giant, but they are small compared to the giant, either 100 or nanometers or 10 micrometers, and these are the ones I'm going to use. And because of this character, this negative character of the microorganism bacteria, we are going to use POPG mixed with POPC. This is the most common lipid in our membrane, and then we add increasing amounts of POPG to have a net negative charge. So with this one, we mimic our membrane, and with this one, we mimic the bacterial membrane. So I'm going to just show some examples here, mainly gomezin. These are already kind of old study, but it's nice to see all these techniques that I showed you yesterday, revealing how this peptide works. Then I'm going to talk a little bit about this one, and this is something that we started now. This is just some preliminary results. So each of these peptides are collaboration with other researchers that provide the peptides. So gomezin was originally obtained from this spider. It's a Brazilian spider. Its name comes from the name of the spider. So once you see that the venom of the spider has this activity against bacteria, you isolate the peptide, and then you get what is the sequence. And knowing the sequence, we can go to the lab and obtain it synthetically. So this is the sequence of amino acids. And this shown here are the charged. So you have 6 positive charges, none negative. So it's it has a lot of cationic character, and also it arranges in this beta hairpin structure. So there are these cysteines that provide a disulfide bond, which is very stable, and this makes that the peptide always stays in this conformation because of these bonds, and it's amphipathic because of this. So it's quite stable, this beta hairpin. That's like this thing that you use in the hair, hairpin. So a beta because it's similar to the beta sheet, and it has this amphipathic character. And we studied also an analog of this, a linear analog in which the cysteine residues were replaced by serine so that you don't form this bond, and then in aqueous solution, this thing doesn't have a preferred structure. It's random structure. So it's a loose conformation. And we use them with our model membranes to see how the activity of each one of these peptides is. So I'm going to call them GM and GML. So first, what we did was to go to the microscope, because it's always nice to see stuff. So we have a micropipette here, a glass micropipette that is injecting the peptide solution. And there is, I hope the video plays. So we come here with the pipette, we blow Gomez into it, and then what we see is that suddenly the vesicle collapses. So we have what is called this carpet mode. I told you in the beginning that there could be either vesicle rupture or opening of pores. So it's a very fast event. Within less than 1 second, the whole membrane structure is collapsed because of the peptide. This is very different from other peptides that work through the toroidal pore, like maganin is one of the most studied antimicrobial peptides. So if we do the same experiment with this other type of antimicrobial peptide, what we see is that the membrane becomes permeable. So we lose this optical contrast, which came from the sugar asymmetry. And so this means that the membrane became permeable completely, but you keep the membrane structure. So there are pores that open through the membrane, but you keep the vesicle integrity. And with this antimicrobial peptide here, we never saw this outcome. So to quantify this, we quantified how much of these bursting events we had. So after some time, we choose a large field, and then we just count the number of vesicles, and then we add some concentration of the peptide, and then we see that none of the vesicles survive, or only one. So what we did was to calculate the minimal bursting concentration, the minimum concentration needed to basically burst all the vesicles. And we did that for increasing molar ratio of this anionic lipid and for the two analogs. So as expected, the activity of the peptide is higher for lower concentration, and then this increases with the charge, because it is positively charged. If the membrane is more negatively charged, the interaction is greater. And if we add cholesterol, the membrane becomes much more resistant to that, which is important because our membrane has cholesterol. So if you want to design an antimicrobial peptide or an antibiotic, we want it to kill only the bacteria and not us. And what is interesting is that if we are at low charge concentration, so there is gomezin has a much higher activity than its linear analog. But if you go to high charge, then the activity is basically the same. And in the end, I will show you how. We also did some assays in which we could quantify this permeability ability, the permeation ability of the vesicles, and for that we use a fluorescence assay on this small liposome. So basically we entrap a fluorescent molecule, and we have a way to see if this molecule goes to the external medium. So when it gets diluted, it changes its fluorescence intensity. So one can quantify the amount of leakage caused by increasing amount of peptides. So we did that for two charge densities, either 25 or 50. And what we see is that the activity increases with the peptide concentration. Now, of course, it should do that, and it reaches 100% eventually. And again, we see that for low charge, so there is a much higher activity of the normal gomazine in respect to the linear one, but at high charge, 50 mole%, the activity is the same. Another way to explore this interaction is to use this isothermal titration calorimetry, the ITC, which is a technique that measures the interaction heat between our liposomes. In this case, we put them in the syringe, and we fill the cell with the peptide, and then we inject consecutively, and then we can measure the heat and create a binding curve and extract the binding affinity and see how this interacts with the membrane. So what we saw here, this is just the integration of the heat as the molar ratio is increased. So for low POPG, GML, the linear analog, doesn't interact with the liposomes, and there is a small interaction with the POPG, with the gomazine. So we could measure delta H and measure also the binding affinity of that. But then if we go to 50 mole%, again, we see more or less the same activity, much higher than with 25. And then the values obtained are also similar, so it's a much higher affinity, much higher interaction energy. And if we go to another technique, which we use in a facility, which is the circular dichroism, this is the technique that can sense which type of structure the protein or the peptide has. If it has alpha helix, if it has beta sheet, if it has random configuration. And what we see is that gomazine, because of this disulfide bond, it's always stable. It's not very sensitive to the presence of so either in buffer or in the presence of the liposomes, it's more or less the same structure. On the other hand, the linear analog changes its structure and starts to acquire a structure that is similar to gomazine once you go to very high surface charge. So when it comes in contact with the membrane, this structures the peptide. This is a very common feature for almost all peptides that are random in solution. And gomazine that is not, because it has these two ridges. So the explanation for what I showed before is that gomazine always has the same structure, but the linear analog is either inactive if it's in bulk or at low surface concentration, or it can become active in a similar conformation once it really gets inserted into the membrane. And something that we observed also, like a side observation, is that these peptides cause a huge aggregation of the vesicles. So it's positively charged, the vesicles are negatively charged, so they bridge the liposome. So this is an optical microscope from the small vesicles. Usually the small vesicles you don't see because it's below the optical resolution. You just see some dots here, 100 nanometers. But once it gets in contact with gomazine, it forms this huge mess of aggregation because of the interaction with the peptide. And we can also measure the size of the aggregates with the dynamic light scattering, so we see that it goes to the micrometer range due to the peptide. And similar to that aggregation between liposomes, we also see something that happens on the membrane of a single vesicle. So the vesicles, they cannot easily aggregate, but what we see are these bright spots here that show that there is a large concentration of lipids there. And these several antimicrobial peptides cause this, and what we see is that the whole membrane collapses itself into this very dense structure. So our proposed structure for that is that the peptide induced, it's like almost like a self-aggregation, it starts to bend, mediated by the peptide. So in case you don't have other vesicle for it to interact, it interacts with its membrane and then forms these very dense regions. Okay, so this was gomezine. Now I'm going to the other one. So this other one was obtained from this frog here. No, actually, it's an analog of a peptide that came from this frog. So there were some modifications. You have an addition of a lysine, and you have the substitution of one of the amino acids for tryptophan, and I don't remember. There were 3 modifications. So this is the peptide that we study, and of course it has also this net charge, in this case only 4, and lots of hydrophobic residues. So this one, we did another technique, which is this one that senses the phase transition of lipids, another calorimetry that I told you yesterday. And for that, we use lipids that have this transition in a measurable temperature, not this POPC, which is close to 0. We use this other that have a transition at 41 degrees. So they are like this alone, and they have more or less the same temperature, so we can either study also mixture of them. But I'm going just to show them separately. So if we add the peptide to this DPPC, if you remember that I showed yesterday, the heat profile is a very sharp transition from the gel to the fluid. It's very narrow. But then as you add the peptide, you start to change, to broaden this transition. So it becomes much less cooperative. So you are for sure interacting with the membrane. But so it's a broadening. But if we do it with the PPG, so you also have a broadening, but you have mainly the appearance of 2 populations. So there is this peak here, which more or less does the same as this one, broadens, but then you have the appearance of this other peak. So clearly you have 2 populations coexisting there. And since this peptide has tryptophan has fluorescence, we can also see where tryptophan resides. So if it's in water medium, it has a certain fluorescence maximum at 350. But as I go to a more hydrophobic medium and with less movement, so we have this, which is called a blue shift, which is a hallmark for insertion in the membrane. So you know that the peptide is entering a hydrophobic medium. And we did that so if the peptide is in buffer, it's in a very polar situation. It's a high wavelength here where it emits. But then if we add DPPC, which is the neutral one, we see a shift. So there is an interaction, but it's not a very strong interaction. And if you add DPPG, even at low lipid concentration or low peptide, all the peptide is bound, and it's clearly in another conformation. Okay, so all the peptide is basically there. So the model we use to explain this is that in the PPC there is a more loose interaction. It's mainly because of the affinity with the hydrophobic core of the membrane. So there is not a very large interaction with the head groups. So you have some perturbation. You can also have opening of pores, but it's a shallow insertion. So it remains here close to the head group. It doesn't go very deep. And since it's not very attached to the head groups, it can diffuse fast on the membrane, so it causes like a global effect on the whole membrane, causing this broadening of the transition that I showed. And for the PPG, since there is an additional interaction with the charges, it keeps the peptides in some region, and then in this region, it affects a lot the membrane. So it can either open pores, or even if it's in an like not open state, it still cause a lot of change in the bilayer properties on a specified region. And this causes this peak at lower temperature to occur. And it goes deep in the membrane. So it's a deep insertion and local perturbation. So we could show how these peptides work using these techniques. And now just to finish, it's something that we started, it's a very early project. It's an interaction with people from Brasilia that make these peptides. So these are called intragenic antimicrobial peptides. So these are natural sequences occurring in our genome, for instance, in much larger proteins. And you analyze the sequence with a software, and then you try to infer which sequences could have some antimicrobial potency, like a region that has this size, has as many cationic residues, and it has this amphipathic characteristic, and the ability probably to form a alpha helix. And you select this, and then you test against bacteria, and then you study these sequences. So it's just, you use our naturally occurring sequences to get inspiration on which kind of sequences to seek for. And so this one is came from, this H is from humans, so it's from it comes from another protein, big protein, and it's a small part of this protein, and it's called HS02. And if you do this analysis here, you show that it has the ability to form an alpha helix in the membrane and an amphipathic one. So the charged residues are on this side, and the hydrophobic residues on this side. And they designed also analogs. So this analog here, you just remove the last 3 amino acids, so to remove the ability to form alpha helix. And then you want to see how the size of the peptide and its ability to form the structures influence its activity. So I'm just going to show very preliminary results. These are ITC results. So the peptides, even though they look similar, they have a very different heat profile. So this one is only exothermic, and this one has an endothermic component. We still don't know what it is. This is really very, very new, but it's very different, its behavior against the lipids. And we have also some optical microscopy to show that it also causes this vesicle collapse. And we are, in parallel, we are working also with the extraction of lipids from bacteria. So we have a collaboration with the people from the chemistry department, and then we take a culture of bacteria, and then we use specific solvents so we can extract the bacterial lipids from this, and then we make these giant vesicles out of this lipid extract. And then our idea is to compare the activity with the synthetic one, but this is still very, very early stage. So I just want to thank all the people present in each of these collaborations, mainly the people who provided the peptide, Antonio Miranda, Eduardo Chile, and Guilherme and Gabriel from UNIB, and thank you for your attention.
This is a very interesting presentation. I'm just curious about a couple things. Are these peptides all amphiphalic peptides, or?
Yeah, these that I mean, in nature you find some that are not, but like the biggest class of them are amphipathic and cationic.
So that means that they attack the bacteria, but they don't interact to sort of for plasma of eukaryote membranes of eukaryote cells and stuff like that?
Yeah, you always, when you do the biological test, you always do with the culture of bacteria and also its hemolytic effect. But in general, they are more effective against bacteria, mainly because of this electrostatic part.
Okay, and just to finish, how much can I make these peptides? I have this question for a long time, and nobody answers me in Houston. How much can I do it instead of just make it better amphiphalic against the bacteria, make less interacting with the eukaryotics? So basically, work on the other part of the design. Do you know what? Any comments on that?
Yeah, I don't know much. So people always look for, they check both. No? They check the interaction with the bacteria and how it's hemolytic. No? So with the bacteria, you go like increasing charge. And, but also if you increase too much, even with the bacteria, it doesn't go much better. It cannot go better. So it has to have this balance between charge and hydrophobicity. So probably, yeah, probably something to do with cholesterol. So if the peptide has a very strong or difficult, cannot, for instance, this one, if you put cholesterol, the membrane becomes more resistant, and the bacterial membrane doesn't have cholesterol. But how you change the structure so that it has less interaction with cholesterol, this I don't know how to reply.
Thank you.
So, hi, thank you for the talk. It was really cool. And I was wondering if you can use the same technique of these vesicles to study protein crowding, the membrane crowding of proteins.
You mean membrane proteins or proteins in the medium?
No, membrane proteins. Yes.
Okay.
So if you just put
so
it
the difficult part is to put these proteins in the membrane, no? Because the way we prepare these lipids, usually we start with a solution in chloroform, for instance, and if you put the protein there, of course, it will lose its structure, and then you cannot easily recover it inside the membrane. So there are ways that you reconstitute a protein in one of the giant vesicles. So there are several people who does that, but usually you put one type of protein, not a very large concentration, so it is difficult to study a crowded system with proteins, membrane proteins. There are people who study like liquid-liquid phase separation inside the vesicle, but this is more like polymer-induced, not protein-induced. But
Yeah, thank you, really cool. And just a second question, like yesterday, if I understood well, so in the beginning of the process, what happens is that these vesicles get like concentric, so you have vesicles inside vesicles, and then you filter it and you get only simple ones.
yeah, it's not filtered, but you rearrange them into...
And can you, instead of rearrange it, if you keep one vesicle inside another, just try to mimic what would be a gram-negative bacteria?
Yeah, the problem is that when you do this, so most of the lipids, when you prepare, they spontaneously form this onion-like structure. So if, for instance, if I go and put this antimicrobial peptide, I will put them on the outside. This means that they will have access only to the external layer. And then if it collapses the external layer, then it will get access to the next one. And each vesicle is very different also, so, and it's really dense. It has thousands of lamella, so it's not very good to work with. Or you can prepare, for instance, if we want to do this DSC measurements, you prepare the whole thing, the peptide with the lipid. And then you see which structure is the one that the whole thing wants to do. And this is another approach. Or I did, for instance, with detergent. With detergent, it was interesting because I saw how the detergent was like peeling off the vesicle and slowly solubilizing everything. But it's rare when we use the multilamellar ones to interact with something that is on the outside.
Yeah, very nice talk, Karin. Thank you. Just a naive question from someone not from this field. What is the horizon or the pipeline between such a nice approach in basic science to eventually this becoming a product of some time?
You mean this antimicrobial
or
peptides?
This or any of these sorts? How is this landscape evolving recently as far as practical applications go? I mean.
Yeah, so we all say when we do the introduction that it has a potential application in this and that, but it's not so close. Even with these antimicrobial peptides, I think there are none still in the market, for instance, available in the market. And the idea to use this now from the model membrane point of view is more to understand the processes that occur in biology. And then if this can help something more specific, then nice. But it's, to tell you honestly, it's more like basic research.
Okay, so let's thank our speaker again. So our next speaker is Professor Paulo Bish. He's from the Federal University of Rio de Janeiro, working at the Institute of Biophysics, Carlos Chagas Filho. So